Hi, good afternoon, and welcome to the webinar about the Master Protocol. My name is Pat Furlong, and online with me is Abby Bronson, our Senior Vice President of Research Strategy, and Cliff Bechtold. Some of you online may remember and have some contact with Cliff Bechtold from the time he was with BMS working on the anti-myostatin program. So with that, we'll begin to talk about a Master Protocol. So first of all, I'd like to think about this as an opportunity for the Duchenne community. And the reason for that is in trials with a Master Protocol, we could expand the inclusion criteria across the trajectory from very young to adults. Most of the trials these days are not inclusive of adults, certainly not over the age of 18. We also think about it as an opportunity to dramatically decrease the time it takes to open studies. And I'm going to talk a little bit about how long it takes to open a study in just a bit. Also to potentially decrease or lessen the time of exposure to placebo. From right now, most of the studies that are randomized controls are 2 to 1, so two patients would be on active compound where one would be on placebo. With a platform study or Master Protocol, those odds could be 4 to 1, 4 patients on an active drug and only 1 patient on a placebo. So certainly decreasing the exposure to a placebo. And then to be reassured to this community that in the Master Protocol, no one would be excluded from a study. So just a story, I had a call the other day about someone who was in a particular study on an EAP, extended access program. They didn't feel that particular compound was providing the benefit they desired or needed, so they stopped that particular EAP, washed out for 6 months to really screen for another study when they found out that they were excluded from the study because they didn't meet the inclusion criteria or no longer met the inclusion criteria. So in a system such as a Master Protocol, you are in the system. So if you completed a study, you could then wash out and go to the next study without ever having to worry about do you meet the inclusion criteria, because this protocol would be consistent throughout. So why? Why would we even think like this? One, because time matters, and I think every one of us who have ever been touched by Duchenne know that time matters, right? We know the trajectory of Duchenne. That prognosis is given to us the day we hear this word enter our family and our lives, and so we know well what is predicted to happen. And I know many of you have been just standing on the outside of inclusion criteria. Not, for example, if it was an 8-second from supine to standing and your son was 12 seconds, you are right outside. Or worse yet, you can't get off the floor. Some have dropped out of a study, as I mentioned, to think about a better study in their view and then been unable to meet that inclusion criteria. And then if we look at time to develop a protocol, time to get the sites selected, time to get those sites up and running, that can be years. So I think that if we think about this as an opportunity, we could essentially really have economies of scale in terms of time. Now, there are concerns, and I know that there's been concerns expressed online, so just to kind of talk about some of those concerns, there is some confusion that all of a sudden one day all the studies would be in the system. It's not true. You build these systems from the ground up. You start off small with a few studies, a few companies that would be willing to enter the system that makes sense to start off the system, and then determine how this is evolving and will it work and answering some of the challenges. We've been exploring this concept for several years. You might have heard us mention it from time to time, and certainly last year in June at our 2017 annual conference, we asked for volunteers from our community to participate in this process to really try to think through what a Master Protocol might mean for our community. We didn't have any foundation volunteers at the time, but we certainly did have a few companies that said they would be willing to work with us and further explore the concept. And why would we do this? Again, just to repeat, we want to accelerate. We want our families with Duchenne to have more shots on goal. We want more people to participate in studies. We want more opportunities, and then to get to combinations, which has been a desire and a mantra of this community since I entered it many, many years back. So we think this might be efficient, inclusive, and results-driven. So I think in Duchenne, one of my worries was always the world I knew would change and become worse overnight. So I just put this slide up there to say we have 29 active protocols. It's a very exciting time in Duchenne muscular dystrophy, and I don't want you all to wake up and think that one day or tomorrow that is going to change. Those protocols are active and they're ongoing, and hopefully many or most of them will get to an approval. But if you think about how these are done right now, each company develops their own protocol, does their own site selection, contract negotiation, and so on. And essentially you can think about it this way. Each company builds a house, they build it, they execute it, they analyze it, they go to seek approval for that house, and then the house is taken back down. So it would be more efficient if we had a big house where we could do efficiencies throughout and save time. So just thinking about how long does all this take? And so this slide just describes to you how long it can take. A year to design a protocol. A year to get the mechanics and identifying sites and doing site contracts and IRB. All of that can take another year. Recruiting patients can take 1 to 2 years, depending on the study, depending on the number of people in that study, depending on the number of sites that are recruiting. And then it can take 1 to 2 years to execute on the study. As you all know, the Essence protocol is 2 years. The Pfizer anti-myostatin is a 2-year protocol. Givinostat is 18 months. So we're looking at longer and longer protocols because of the fact that Duchenne is a slow-moving disease, fast in our hearts and eyes, but a slow-moving disease in terms of trying to measure it. So these trials are longer, and then it takes several months to lock the data and analyze it, and then there's a process toward approval. So all of this, on average, can be around 6 years. And so I just pulled off clinicaltrials.gov a little bit of information so you could see. On your top left, if you're looking, the PS06252616 is Pfizer's study. You can see that it was first posted December 8th of 2014, and this was when their first site was open. This didn't talk about the preparation and site selection and all of that. This is when the first site opened, and it's scheduled to be completed in 2023. So you're talking about a number of years. Fibrogen is another one. Start date in 15, completion date estimated in 21, 6 years. AVI, as you know, AVI that turned into Sarepta, started in 2009 with an approval in 16. So we're talking about years of time when we should be thinking about getting to combination therapies. The Essence protocol is the one on the left, the SRP 4045 and so on. Again, posted in 15, end date is 2021. The NS Pharma is below that. Talk about when that started. April 15th of 16. It is going to be completed in 19, so that's 3 years, and that is a dose-finding study where they're going to have to then do a phase 3. So that's going to be at least 6 years in development. And tamoxifen, again, I know a little bit about the history of this. Elizabeth Vroom in the Netherlands began funding this research for many years. PPMD added on to the funding of tamoxifen and is now identified for a clinical trial funded by Duchenne UK, which is terrific. November 17th this was posted, and completion date is 2020. Again, time. We also looked at data to say, what do the patients and families want most? And so this is a survey that we've done with SMA, but Ryan kindly cut this down so you can see a little bit, I'm going to go back, sorry, about what this is and what was most true for you. Most of the people, about 68%, wanted their children in clinical trials, but there was 34% of these individuals who said they did not want to participate in trials. So I know we have a very engaged community, but we have to respect the fact that there are 30% or more who aren't as engaged and perhaps not as willing. In terms of neuromuscular provider, we asked how much the clinicians talk to you about this. So how much do they tell you about these trials? And you can see here that 50% of them thought they had some good knowledge of trials, but when you look at the middle one, how often does your doctor talk about research opportunities in advance? 68% of people said never. So that's something to think about in terms of who is participating in trials. And the next one is, in my opinion, my child's doctor thinks my child should be in a trial, so that has pretty good references in terms of that 76%. But, only 37%. But I have no opinion is 57%. So clearly, the engaged community is in trials, but there is certainly a segment that is not. What is the biggest concern? the biggest concern of the community we see over and over is my child could receive a placebo, the inactive medicine, and how long could my child receive that? So this is an overarching theme. No one wants a placebo or a saline infusion. I'd be more interested in putting my child in a clinical trial, and you can see the results here. I won't read them, and these will be posted. If I was confident the trial would improve researchers' understanding, and at the bottom you'll see if I knew my child would receive active drug and not placebo. So these are recurring themes that we've had. In the preference studies that we did, and you'll recall that we did a preference study in 2013 and presented it to the FDA. The highest priority found in that early preference study was slowing or halting disease progression. Later, in the policy forum of December 2013, we heard again the risk of placebo, time, and exclusion criteria based on age or function. Again, in the draft guidance that we did and submitted to the FDA in June of 2014, we had the community advisory board that consisted of more than 100 people in this community, and their voice turned out to be the imperatives in a cover letter for Dr. Woodcock. Again, these themes were repeated: inclusion of all people, clinical milestones that are measurable across the trajectory, limited exposure to placebo, and take into account the benefit-risk equation of families. So we've been thoughtfully thinking about this for a long time and having conversations with the community in different ways at different times. And what we are doing now with a team of people is exploring this, asking ourselves what are the challenges, what are the questions that need to be addressed, and thinking through if we could get these efficiencies, if we could get trials up and running within a very short period of time, if we could have patients across the trajectory of this illness participating, if we could do things quicker, better, get to approvals faster, wouldn't that be worth a try? So Cliff is going to take it from here. Cliff?
Thanks, Pat. What I thought I would talk about today was really to cover and address what is a master protocol. It's thrown around a lot, and I think there's an opportunity to be clear of what we're trying to build here when we think about innovative adaptive design. And I think just to clarify, though, that we're also at the very early stages in this. Master protocols, or some of the different forms of master protocols, have been used in Alzheimer's, oncology, and very successful. And we're trying to see if there's a way that we can adapt that to work within the Duchenne community. We're at the early stage. We're looking for putting a structure in place that we can flesh out, as with some of the key questions that we are putting around this. And we'll be working with the entire community, with the patients, the investigators, site coordinators, sponsors in the industry, and the clinical research organization to seek their input as we continue to determine how to best bring one of these innovative designs into the Duchenne community. And so the question is why? Let me just start off with what we're trying to achieve. So in innovative adaptive design, or master protocol, there's often three different types of master protocols that are considered. A basket design is a single study with one drug, but there's many different patient populations. You can think of this as, what if we were going to do a study, we had a drug that helped overall muscle health. We might want to study that drug in Duchenne, we might want to study it in SMA, we might want to study it in Becker's, and we might want to study it in ALS. So we could create what's called a basket design study, in which we have 4 arms of those different muscle degenerative diseases, along with one placebo, and simultaneously we can study that one drug in these different groups. And that would be called a basket design. An umbrella design of a master protocol is one where we look at a single population but study multiple different drugs simultaneously. An example of an umbrella design, you could take, let's say you had an anti-inflammatory drug, or 3 different types of anti-inflammatory drugs, I should say, and you want to study those at the same time in the Duchenne patient population, and you want to have one common placebo. So you start enrolling into all of those 3 drugs at the same time. Once you have enrollment, you would stop enrollment and go into the analysis and continue on from there. What we're looking to bring into the Duchenne community is a platform design. And this is a single patient population, being Duchenne, with multiple different investigational agents that can be compared to placebo, similar to an umbrella design. But here it's in perpetual manner, meaning that not all the arms and not all the drugs that come into the study start at the same time and nor do they end. And I'm going to give you an example of that in a coming up slide. But the reason that we think it's important here is that by bringing in a platform design of a master protocol, we think that it's an opportunity to broaden into different patient populations or go into wider. So we could include that not just the most common age group of the ambulatory, but we could also include a broader group into the non-ambulatory. We think we can improve the standardization around the endpoint, not only the selection of that, but also the training and the data collection. We think we can streamline the site engagement and recruitment to optimize the limited resources we have. Many of our investigator sites have limited resources within their study coordinators, and all the different studies that are coming into this space are competing for those resources and competing for the number of patients that would be enrolled. If we can create one protocol, we can simplify standardizing around these endpoints and data collection and how we actually execute the study while we use a common placebo and thereby minimize or limit the number of patients that are actually exposed to the placebo arm. This is not, as I said, it's been used in other areas, and Janet Woodcock led a paper in the New England Journal of Medicine last year that summarized the different types of master protocols and really highlighted the innovation by using the conservation of the infrastructure from the Data Monitoring Committee, from the IRB, the trial network, and also by using an adaptive design, how you can continue to improve the execution of your study. And that's why we are looking to bring this innovation of what's being done in other therapeutic areas into the Duchenne community. This table, or so this graphic was taken from that paper that Janet had last year, and this describes what a platform design of a master protocol would look like. Patients are screened into the one study, and then they, based upon their entry criteria, they could be randomized to different subgroups, and what's used is called stratum here. But there could be a collection of boys that are based upon their ambulatory status. That we could have a grouping based upon a genetic marker. There's different things that we could use, and in our case, we're looking to bring a study design that would allow both ambulatory and non-ambulatory as two different strata. And with this example here, once they're into that group, so let's take a look at biomarker A strata, they would come in and study would start, and there would be two investigational agents compared to a standard of care, which could be placebo in this case. The patients would be enrolled. The study would be predetermined on how it would be executed. And as you can see in the example here, that investigational drug 1, by how it was probably designed, hit its criteria, was met. It would go into analysis, but investigational drug 2 would continue on. And as time progressed, maybe there's another investigational agent that would come in, and in this case, it's investigational agent drug number 5, which is then entered into that stratum, and the study continues on. Meanwhile, we take the learnings that we got from the first part of that investigational drug 1, and now we update the protocol to reflect how the things have changed over time. If you look at the example in the middle with biomarker B, in this case, there was an investigational drug that part of its predetermined execution plan was to look for futility or show that determine is the drug not working. And in this case, it would hit that would be terminated, and the patients that were enrolled in the study now would be eligible to continue through a washout period and re-randomize back into the study. So that's an example of how when you hear us talk about a master protocol, what we're really talking about is a platform design of the different types of master protocols. And this schematic from Janet Woodcock's paper in the New England Journal of Medicine shows what that would look like. So moving along, why do we want a platform or master protocol? this has already been demonstrated that these novel designs have the ability to minimize those requirements. It's what you've heard, some of the friction points that Pat had mentioned, but it's, we're in a rare disease space where we can't do the traditional large number of studies with a large number of patients to come in to fulfill all those different studies. Here's an opportunity that we can make the best out of the resources that we have in a way to make sure we're being most efficient with the patients coming into the study, and that their data that we are getting from the boys that are coming into our study we're getting the full benefit of that in our analysis and design the study. We really think we can shorten the development timelines. As Pat went through all the upfront time, trying to get MRIs qualified and other equipment qualified for each individual study, we could just do that up once. And as other new drugs come in, as the example that I had in the prior slide, in this slide here, all the protocols already going to be approved as the time that investigational drug 5 would come in, and therefore it would not have to go through that long and lengthy startup time. We think that there's a way to consolidate a lot of our learnings. If you think about how we do drug development, every company that comes into the space has to learn from the Duchenne community how to do a study in Duchenne. And so they go to the health authorities, both in the U.S. and Europe, and they get their feedback. So every company is doing that individually. What if we could consolidate that, get feedback, and then share that across the community? Every individual company also goes out to all the thought leaders and investigators to understand exactly how do you perform, or what is the appropriate testing that we should be doing looking at the efficacy and safety of these drugs. We could bring that together and make sure that we're using the best thought and contributions in how we design the study. We can also consolidate our data around what we know about natural history data. So natural history data is understanding what is the normal progression of a boy with Duchenne. And we can understand how they perform on some of these endpoints that we use in the study, and we can share that across and make sure we implement that back into the design of our study. But we can also have a more powerful design and use of statistical adaptation and inference in how we further evolve the protocol moving it forward. And we think we can achieve all that while maintaining something that's an advantage to the companies. I mean, the companies need to do their own analysis that might be specific for their mechanism of action. They might have certain endpoints that they want to do that might be relative just to their molecule that they're studying, and they might have a different hierarchy of what they're looking at. We can maintain all that confidentiality and provide that to the investigational agents that are coming in while maintaining all of their learning. This isn't going to be easy, and I think it takes us coming together as a community to make it happen. And it's easy to throw up all the hurdles of why this isn't going to work. The challenge is to find a way to overcome that, and that was part of my journey in this. Is that when we first started having a conversation, I thought there's no way we'd get this off the ground. But once we started looking at all these hurdles and spending our time thinking about how to overcome those hurdles, we all of a sudden started to see that, hey, this might work, and we might be able to get the benefits from it. Moving on, the objectives of the master protocol, just to reemphasize it, looking at that optimal execution and data collection, the safety monitoring, if we can streamline the burden to the sites, the investigators, the families, and then shorten that overall time from that Pat had shown in the timeline up front, the contracting, the protocol design, and the training that goes into this. If we can also standardize and for the patients as far as what their experience is, and make sure we have the best practices of an informed consent, make sure that the training and onboarding of patients into the study that were somewhat uniform across all the different studies, that we can standardize a lot of the testing and home administrative activities. These are things that we feel that we will be able to achieve as we work forward in creating this master protocol. And just a few slides here, just as an example of what the evolution that's going to happen here is that this is the current process where every single in clinical trial A, B, and C, as Pat had gone through with the timeline up front, all these individual studies need to go through IRB reviews, site agreements, set up your database, create your own informed consent, work with the site to get them aware of the study, make sure that they have the capacity to do it, and then training, often involving travel away from the study site. And then once you get up and running, you have patients that are coming into each individual study, get randomized to placebo versus their active drug. What we hope to see in the future is the shift to a master protocol, where up front, we do it once. We do the IRB review, we do the site agreement, the database, we create that. And once that's in place, as a new drug comes into it, all we need to do is update that piece of it. We don't need to start from ground zero. We just need to build on, and the site only needs to really review the safety and appropriate information for that new molecule coming in, not the entire program. And then patients coming in can get randomized into these individual arms, but that would eventually lead us into a place where we can then adapt and take the learnings from that and optimize that patient's journey through the program, trying to get to an earlier decision on drugs that work or may not work, and then hopefully get into a place that we can actually look at other things such as combinations of these different agents within a particular patient population. So that, in a nutshell, trying to put some text around what a master protocol looks like, and now I'm going to turn it over to Abby.
Hi, everybody. I'm going to talk about the process and what we're doing to try to design this thing. And I think it's important to understand where we're going, to sort of take a look back at where we've been and what we started doing. So our first initial dive, as I call it, was back in March, where we had worked with the steering committee of our Duchenne Drug Development Roundtable to try to figure out their priorities and some issues that they wanted to work on. And keeping in mind the imperative that Pat mentioned at the beginning of the talk, the Drug Development Roundtable Steering Committee wanted to work on clinical trial optimization. And with that, meaning looking at some of these adaptive trial designs that people have heard about, the Bayesian statistics underlying these trials that allow this type of trial to take place. So the agenda is kind of small. I apologize for that. But we did have a session on adaptive trial design. This was our first initial regulatory engagement. Lisa Lavange, who was the other author on that paper that Janet Woodcock that was published last summer, she came and gave her perspective, the regulatory perspective on all of this. And we had Jeff Allen from the Friends of Cancer, because lung map has been done in that in lung cancer, and so we heard about that trial design. So it's a really opening, very interesting session for us to hear. And I was just looking at this agenda and remembering that Ed Kay, who was the CEO of Sarepta at the time, he presented a platform trial for multiple exon skipping drugs within the same protocol. So essentially a mini example of what we're talking about now. So then from the time of that meeting where we were really interested in this idea to the Connect conference last summer, we really spent time educating ourselves, just talking to other people that had done it, experts, biostatisticians, regulatory experts. We continued with the regulatory interactions, but we were really wondering, how would we do this? And we decided that we needed to put together some sort of a team to take a good look, kick the tires, if you will, as to whether this is even possible in the Duchenne space. So last year at the Connect conference at our Drug Development Roundtable, we revisited the master protocol concept, and I did ask for volunteers for a group to, quote, kick the tires. And as Pat said, I did feel a little lonely sometimes because it was hard to get volunteers, but we did get a group together. We started meeting once a month, talking about the issues, and as Cliff said, it was really easy to say, oh no, this can't be done. But when we took the time to think about, but look at the benefits, is there a way to accomplish this? I think we began to see that there could be ways to do and to meet those imperatives of efficient, inclusive, and results-driven. So we had another drug development meeting in October, and we decided to continue, and that this was really something we needed to take a deeper dive into. And I put this slide up because meanwhile, after that paper that was published in the summer, the Janet Woodcock and Rissa Lavange paper, there was so much press on this idea of adaptive trials and master protocols. It's in some of the PDUFA 6 language and the regulations. So there clearly is a lot of momentum for this concept. So after that October meeting, we decided that we'd form we switched the nomenclature from exploratory to design, because we thought maybe the best way to do this was to put together a Strawman protocol and then bring the entire community around it to say, yes, this is going to work, no, this is not going to work, what about this? Get all the input, because it's hard to react to something when you don't really know what it is yet. So we formed this design team to really look at what was possible. We did need to formally engage a biostatistician because that is expertise that nobody from industry that we were working with had, although I'm sure there's plenty of people in industry that could do it. But we had our own. We engaged with a biostatistician expert. We had regular meetings, and we set out a series of questions, very fundamental design questions that we knew we needed to answer to be able to move forward. And we're targeting Q4 to present the initial findings. We're going to have a public workshop where all stakeholders are welcome to come together and listen and provide feedback. And all along the way, we have had great regulatory feedback. Our next touch point with the regulators is on May 10th, and that's just to make sure as we think about these questions and come up with what we think might be the right answer, are we going down the right path? What could they add? And I just wanted to take this opportunity to say thank you to this design team that has been working with me very diligently. I listed them out here, and they have just been so helpful in answering these questions. And I think it's a great group, and we've really come to value each other's input and working together a lot. So just wanted to say shout out and say thanks to them. I also thought that it would be important to point out some of the characteristics that I asked for back in, I guess it's probably November or December, when I was changing from the exploratory to the design team, because I think it represents where we were in the process and what we were trying to do. So it was a serious commitment. I mean, it's not going to happen by itself. Like, we really needed to roll up our sleeves and come together, and they had to commit to a conference call every other week. And obviously, we are designing something for the future. We don't know what compounds are going to come into this, but we want to design something that can last and is built to handle whatever is going to come in. So I needed to find people that were open to considering this, but they didn't have to commit to having a compound at that time, but they would consider it once the design was up and running if we got there. And I needed clinical trial experts, people who were really involved in that aspect of designing trials, and also came with an open mind. As Cliff said again, not looking for ways that we can't do this, but seeing the benefit and helping us achieve the goal that we want to achieve. And I have to say that I got the A team, so thanks again. So where do we go from here? Our goal, as I said, is to have a public workshop in the Q4 time frame that will have all the stakeholders present, clinicians, the community, industry, the regulators, and really kick around a Strawman proposal there and decide if this is a go or no-go. In the meantime, this design team is going to continue. We have to come up with some answers on the design and some endpoints. We've created very recently, we'll be looking for volunteers, so if anybody from industry is on the line, you can look for an email. We want to facilitate some additional data points, so we're having to do some research to get there. So we've got work streams in an MRI, cardiac endpoints, PROs, and wearables. We'll have to engage with some of the other stakeholders, such as clinicians and CROs. And how can you become engaged? As I said, this is sort of the next big push for us, is to engage with other stakeholders. And so what we want to do is form a patient advisory board and hold community town halls at periodic times leading up to this the gathering, the public workshop in the fall, where we'll be able to receive input and talk about the progress that we've made. So that's where we are with the process and what we've accomplished and how we think we're going to get to where we want to go. And I think we can open it up for questions now.
Yeah, thanks, Abby, and thanks, Cliff. I so appreciate all of the work that you've done, and certainly your design team has been really aggressive and thoughtful about moving this forward. So I don't know if anyone has questions. If you do, we have about 50 people online, so it may not be the best idea to open all 50 lines. But if you do have a question and you'd like to put it in the little feedback where it says participant feedback, it would be helpful, and we could certainly discuss this. As Abby said and Cliff said, we are moving into trying to really sort through thoughtfully some of the questions and challenges we have. Dr. Woodcock and colleagues feel that for rare disease communities, this is a very good approach and one that would likely lead to better outcomes and certainly faster trials and better outcomes. So we feel that this is an opportunity for us to really consider and as Abby said, to kick the tires to see if this could speed things up. We do have a couple questions coming in. So from Lisa, I'm a stakeholder CRO. How can I get engaged? I would like to join the public workshop, but I'd like to be engaged sooner. Is there anybody I can contact? Yes, Lisa, send an email to Abby at parentprojectmd.org, and that will connect you directly to Abby, and we'd certainly love your help. Who's going to run the master protocol? That's certainly an interesting question, Tiffany. I'm going to ask both Abby and Cliff to comment on that, because ownership is not. This is really a community approach with many stakeholders, in particular the patients and families. So I wonder if you wouldn't mind commenting, Cliff, and then Abby.
Yes, thank you, Pat. I think that the sponsorship of this and what we know from other areas, it really does take an organization of the patient advocacy group coming together, as seen with the Diane Network in Alzheimer's and in the oncology space. It helps to bring the key people together from the industries and bring them to the table to talk about the design and bring forth the execution. But to execute this, you don't need necessarily that large of an organization, since most of it will be outsourced. So the question earlier on the CRO, a lot of the execution from the overall creating the database and execution of the day-to-day will be outsourced through a CRO. We will need to have, and as part of our planning, creating an organization that will serve as the leadership team to drive and facilitate that execution. Along with the CRO, the providing the drug and the things that would typically go from the drug supply, the sponsors will be actively involved with that in how they source it for the individual study, but we will be looking through predominantly outsourcing and engaging other partners. And maybe, I don't know if you want to expand upon that concept of a leadership team partnership.
Yeah, that's exactly what I was going to say. I think that, you can talk about who's going to sponsor it in terms of the strict definition of the, legal sponsor from running a trial, but also the ownership in terms of we have to put some governance in place, and that's going to be made up of, representatives from all the different stakeholders. And I would say that they're really the owners of, the question was who's going to run it. They'll be really running this trial.
Okay, thank you. Jody Wolf asked about the current limitation on trial sites. As we all know, the trial sites that we have in the U.S. are pretty overwhelmed with the number of trials. And so I'll make the first comment, and then I'll ask again Abby and Cliff. So Jody, it's clear we could use some more trial sites, but I think part of this is all of the other activities that go toward getting the site up and running. So I would be hopeful if you could be more efficient in some of those ways. And also in each trial, as I mentioned, each company builds a house and then takes it down. So if you have a master protocol, then I would think that the burden would be less on the clinical sites. And I'm not arguing the fact that we do need some additional clinical sites, but I'm saying that a master protocol could essentially decrease the burden on the clinical sites. Cliff, Abby, do you have thoughts?
No, I would agree, Pat. I think, I would really hope that this would. And Pat's heard me say that even if the master protocol doesn't achieve everything we hope it to achieve, if at the end of this we come up with, a best practice of how to execute a clinical study so that company X is doing the testing in the same way that company Y is doing it, it will make it easier for the sites, and we'll have, they won't have to worry about, well, gee, in this company, do I do 4-step first, then 6-minute walk? They'll know that this is the pattern they go through, and they won't. I think we'll have better quality of data. We'll have less lost data because it wasn't done in the right sequence by however company had executed it. So I really think that this is going to benefit either in the master protocol or even as other protocols copy what's put into the master protocol. I think it will be a very benefit to the operation of the site, which, as you point out, have such limited resources. And I think it'll make it easier just to expand to other sites that may not currently be in our network.
Right. Thank you. Abby, thoughts?
Yeah, Pat, Cliff said that's exactly my thoughts, what she said. I think there's, sort of a clear regulatory pathway designed up front so that we have this protocol. And as Cliff said, even if that's what we achieve, I think that will help, relieve some of the burden at sites and extend to other sites.
Great, thank you. Then one of the questions is, what's the FDA stance on the requirement of an active placebo arm? I can say that the model that the FDA likes, and I'll have Abby and Cliff comment, is they like placebos, randomized placebo-controlled trials, because they feel that that's really important. I would say that if we could get, over time, if we have a very accurate natural history, which, as we all know, that no two 8-year-olds are the same, no two people with the same mutation are necessarily progressing in the same way. So if we understood some of the variables and biomarkers and genetic modifiers that are changing the progression of this disease, we may, over time, be able to avoid a placebo potentially. But Abby, Cliff, do you want to comment?
Yeah, I think that's right, Pat, and the FDA is being very flexible and realizing that in rare disease patient populations, sponsors don't have the data needed to design a trial without a placebo arm. But they're looking for ways, and they had a whole meeting on it a couple weeks ago, looking at ways you can borrow previous data. So they are being flexible. But until there's really great natural history data, I think we'll have to have some sort of a placebo. Cliff, would you agree with that?
Yeah, I think they've got their stance on using natural history as a control, so I agree with you.
And Tiffany, I'm going to take your second question first, and then I'll go back to your first question. Who hires the CRO? This would be the committee, the steering committee or executive committee or whatever it's called, that is developing the master protocol, or that is leading that master protocol. So that's who would hire a CRO, and I'm certain that there would be some inherent knowledge or certainly exploration of who best would serve the master protocol. So I think that's an unknown. It would be left up to those who serve on the committee. Then will the patient be able to decide which drug they receive? So that's always an interesting question, right? Because we go into trials after hearing about the mouse data and the dog data and whatever other data is available. And I think as parents who are really engaged, then we believe that something is going to be best for our children. It could be, and maybe, I'll make an assumption here, maybe we believe that today that anything that replaces, expresses, or otherwise delivers dystrophin, or a different protein into the cell is where we want to go first. It could be that those studies are independent studies. It could be that all the downstream or some of the downstream would be here. And so it wouldn't be a mystery drug A, B, C, D, and E, right? You would know what companies were in there and potentially and their targets. So I think this is a question, and again, not all studies are going to be within this protocol. The idea is, can you really execute and accelerate some of these other studies? Because if theoretically, if we all were right and said if you express dystrophin to 5%, that's pretty terrific, and then you wanted to build muscle or decrease inflammation or improve the mitochondrial function, as a combination, this is the fastest way to get there, right? So I think this is a question that we really have to talk about as a community and think through, and certainly the design team is thinking about that. I wonder, Cliff, if you could comment.
No, I agree. And agree in that there's different ways that you can design this, and there's a trade-off for every change you make. So if you made it where your most robust study design and statistical purity in how you can execute the study, you would be as agnostic into that decision as you can. You would want to randomize them out. But I think that what's important here is we're in the early stages trying to determine what's best for this community, and that's why a lot of the meetings that we have coming up to get input from other sponsors, patients, and investigators will help us to determine what's the best structure that's going to realistically work with this community. Abby, you want to build?
Yeah, and I think that's a really valid question, and I can understand how a parent would want to make that choice. But that assumes that you want the world of clinical trials in Duchenne to stay the same as Pat, kind of depicted at the beginning. And I think that I think it's a trade-off. But and again, not all products and not all sponsors will want to participate in this. So I think, and another way to look at it is because by creating a clear regulatory pathway, we might make Duchenne a more attractive space for sponsors to come in. And that could even mean there could be more choices for patients at the end of the day. So there's a lot of, it's a complicated question to answer because there's so many different angles to look at it from.
Well, I think you can also look at it from the point of view of a very small company who doesn't have the resources to build a house and execute on the house, right? So if they could come into a system such as this with basically efficiencies built in, then that might be also an advantage for that particular small company or small companies. And Ted Abrish has made some comments. I don't know if you can all see them, so I'll just read them. As a former clinical site director, and as you know, Ted Abrish, or you may know, Ted Abrish has been working with Craig McDonald and the Synergy Group for a very long time. He said this would minimize work with the IRB, contracting, and training. We're trying to implement as many trials as possible without losing money. Difficulties will be ages in the study, determining proper outcome measures or multiple measures, and differences with regard to progression, ambulation, pulmonary, cardiac, etc. Would it be best to include very young, but no outcome measures yet? So I think these are all questions for the design committee as they develop this Strawman Protocol. Is this at first for very young kids to go in and look at the role of inflammation on them? And I'm not suggesting that's what it will do, or this is adults, right? This is everybody over the age of 18 so that we can look at other things. So again, the design team is trying to think about how to best serve this community and get to the answers that we need so desperately in terms of combination therapies so that every single individual with a diagnosis has opportunities to participate and opportunities for therapies and therapies to reach our highest goal, which is stopping disease progression. Cliff, Abby?
Yeah, no, I think I agree with the comments there. And Ted, you hit right on the topics that we spend many of our Friday afternoons talking about because, as you know, it's going to be difficult to come together on all of those that we think are best. But we feel that there probably will be about 80% of that we can align on, and then that 20% we might have to have some flexibility into how each sponsor is able to collect that data or analyze that data. And in the end, we're going to have to come up to a place where we think is the most realistic and best for the community to execute on, and reaching consensus on that will be quite difficult. But I think the other point that you highlighted, which is so important, is to include a broader age group into this. I mean, right now, most of the studies focus on a particular age range and performance level within the community. If we do this right, we could actually expand that and to include even those that are younger, have access to an investigational study, and those that have reached a point of non-ambulation, that they might have an earlier access to some of these investigational agents, both critical to help us advance.
Great, thank you. And again, this isn't tomorrow. I just want to reiterate that it's not like tomorrow you're going to wake up and we have a master protocol in place. To be fair, I wish it were quite that easy. But this is for how do we get someplace faster, better, and this is something that's in development. So this is not tomorrow, not 6 months from now, but we're working really hard to develop the best possible opportunity, and then to explore that with the community in the fall when we have an open public meeting where you can kick those tires. And then together we can say, as a community, we think this is viable, and move it forward. Because if it can solve some of these efficiencies, then we can get where we need to go faster. And Ted, I'm going to take your comments as your willingness to volunteer, so I hope you'll reach out to Abby and volunteer, because we would love to have you volunteer. And anyone else on the phone who is interested in helping us, we never thought this was easy, but we thought it was incredibly worth trying, because the people we love with Duchenne and those who are on the phone with Duchenne are the people we really want to achieve that highest goal, stopping disease progression. So I don't see any more comments from anyone. I hope that all of you, and again, this will be posted so you can review it as soon as in the next day or so. And I hope that you'll review it. I hope you'll really think through what we're trying to accomplish. I hope you'll see it as an opportunity. And then please reach out to Abby and or any of us and say, I'd like to participate, because you know, it takes a village. I think all of us with a diagnosis and all of the physicians who take care of our sons know that we are a village and we can do really good things and have. So I thank you for joining our webinar. Please let us know if you have questions. Volunteers are needed and welcomed. And thank you again for joining us. Thanks, Abby. Thanks, Cliff. It's always a pleasure. And thank you, Joanne, who's been with us developing the webinar all along the way. And thanks to the design team as well. Thank you again. Hope to see you all soon.